Okay, good morning everyone. Good morning Priya. Oh, what's this? Good morning Saima, good morning Jayashree. Okay, let's start. In the last class, we were discussing about the good morning antigen and antibody interactions, is it? So friends, let's quickly understand what we have finished so far in the immunology chapter. So friends, so far what happened? Okay, one second. Yes, sir. So far we have completely studied the cells of immune system. We studied. So after that, we started studying about the good morning everyone, good morning. Okay, later we started studying about the antigen and antibody interactions. We have studied. After antigen and antibody interactions, no, I think we have studied only this. Cells of immune system, then followed by the antigenicity, immunogenicity, and antigen and antibody interactions we have studied. So now we are going to study the most important topic, that is the MHC. See friends, from this particular MHC concept, we can expect experimental based question also. We can get statement based question also. Okay, so far we got both type of questions in the exam, and mostly friends, we got the question from the statement related. Good morning everyone, continue. Most of the time they use it to ask the question about the differences between the different type of MHCs. Majorly they use it to raise the question on differences between the class 1 and class 2 MHC molecule. This is the question they use it to ask more commonly from this particular MHC, and it's very important concept friends. I want every one of you to understand very clearly. Okay, listen, let's focus. MHC molecule. So listen to me friends, when you look into this particular MHC complexes, all of you understand the MHC complexes are not present individually. Let me explain you 20 minutes. I'll explain elaboratively, just pay attention. All the MHC molecules friends, can be arranged in the form of a group, can be arranged in the form of a cluster. So what we can say that the cluster of genes, or are friends the group of genes, which we basically called it as friends MHCs. And if you ask me one common point, if you ask me one common point or one common function, what is the important role of the MHC molecule? I can tell you very clearly that MHC plays a crucial role in recognition and majorly involved in the discrimination or differentiation between the self antigen and foreign antigen. This is the ultimate goal of an MHC molecule in our body. So friends, listen to me very carefully. If you look into the cluster of genes or a group of genes of MHC, they are majorly present in the chromosome 6 in our body. But friends, when you look into the mice, mostly friends, this cluster of MHC molecules are present in the chromosome 17. Please kindly don't confuse. Most of the time friends, instead of using the word MHC, good morning, they will use the word HLA. So human leukocyte antigen. In case of a human body friends, instead of using the word MHC, we will use the word HLA. Whereas friends, in case of a human, in case of a mice, instead of using the word MHC, we will use the word H2 complex. I want you people to remember these names compulsory. H2 complex in mice and HLA complex in the humans. So friends, listen to me carefully. Lot of confusion and lot of information to be remembered. Focus very clearly. Friends, there are total 3 classes of MHC molecules are there. One we can call friends class 1 MHC, class 2 MHC, then followed by the class 3 MHC. All of you listen my statements very carefully friends. class 1 and class 2 MHC molecules are, good morning everyone, continue, they are 90% structurally as well as friends functionally similar. Which one? Class 1 and class 2. But everyone keep it in your mind friends, class 3 MHC molecules are, they are not at all structurally or functionally similar with this particular class 1 and class 2 MHC molecule. Kindly do not get confused with this particular point at all. Okay, now let's look into a small experiment with respect to this particular MHC molecule. Everyone pay attention friends, everyone look into the picture very carefully, don't get confused here. This is one parent friends, this parent having the MHC, the type of MHC we will be calling it as friends BB type of MHC, simple point. And friends, you can see this picture, and this is called as what friends? Good morning, good morning. This is called as a KK type of MHC in the another parent. Friends, all of you pay attention. These two parents giving rise to the progeny in the F1 generation. Everyone please listen my statements. In the F1 generation, the progeny having the type of MHC which we generally call it as friends BK type of the MHC molecule. Please kindly do not get confused with this particular point. Once again understand, one we can call it as BB type of MHC, another parent having the type of MHC called as KK type of MHC. When the two parents in the F1 generation, they are giving the progeny which is having the MHC of BK type. Now you listen, understand the concept very carefully. Friends, you know right, I told you in other lessons also, especially during the organ transplantation, our MHC molecule should recognize that donor DNA as the healthy donor. Suppose example, the MHC molecule present in our body recognizing the donor DNA as the foreign DNA, what happen? Tissue resection may happen. So that's why friends, sometimes what happen? Listen my statements carefully. Sometimes even though we are taking the tissue from the healthy donor, suppose example, listen my words, I am undergoing the liver transplantation. During the process of liver transplantation, I am taking the liver from a very healthy donor, my close relative, but still sometimes what happen, my MHC start recognizing that healthy donor tissue also as a foreign tissue. That time what we will do, we will give the immunosuppressants and we will activate the T suppressor cells and we will suppress our immune system. Listen to me very carefully. See, this is the parent and this is the one which is acting as what friends? This is the one generally acting as the donor. Now, this particular donor, to whom it can donate this particular tissue? Friends, it can donate the tissue only to the BB parent. It cannot donate the tissue to the KK parent at all, because this donor having the type of MHC BB and the recipient having the type of MHC KK. So that's not possible. It cannot donate it. At the same time, friends, keep it in your mind, this BB parent can donate to the BK progeny recipient. Why? Because we understand very clearly the progeny having the MHC type of B type also, the progeny having the MHC type of the K type also. So it can easily receive the tissue from the BB parent. Kindly do not get confused with this particular point. Now you look into the second case, friends. If you look into the second case, who will be the donor? The donor will be the KK parent. Correct na? So friends, to whom it can donate or from KK parent who will receive? Undoubtedly friends, the KK, I mean to say, another recipient who is having the KK type of MHC, it can easily receive. But what about the The recipient having BB type of MHC, it cannot receive. At the same time, the progeny can easily receive the tissue from the KK parent, because the progeny having the K type of MHC also. Now you look into the third condition. Don't get confused, friends. In the third condition, who will be the donor? The donor will be the progeny. And you already seen that the donor having what, friends? The donor is a B and K type of progeny. So you have to listen. Who can receive from this particular BK progeny? Friends, from this particular BK progeny, only the recipient who is having BK type of MHC only. The recipient who is having only BB type of MHC, it cannot receive. The recipient who is having KK type of MHC, it cannot receive. Only the recipient who is having the same MHC composition, that is the BK type, that only can receive. So friends, with this small experiment, what is the conclusion that we can give? We can give the conclusion that during the transplantation, both the donor and recipient MHCs should be matched compulsory. If not, there are many number of chances are there that the organ transplantation or tissue resection can occur very faster. That's why I can give a final conclusion that MHC molecules in our body plays a crucial role in discrimination between the self and non-self antigen. Between the self and non-self can be recognized by the MHC molecules only. Okay, listen my statements carefully for 10 minutes. First, let me explain you, friends, the structural differences between the class 1 and class 2. Then I will explain you the functional differences. Okay, so listen to me carefully, friends, for 10 minutes without having any kind of a confusion. Just listen to me carefully. And I have given a point that both class 1 and class 2 MHC molecules are what, friends? Structurally and similarly 90% they are almost similar. Is it? That is what the point I said to you. And friends, listen to me very carefully here. What is the ultimate goal? Ultimate goal of this particular MHC molecule, the goal is common. That is recognizing or discriminating between the self antigen as well as the foreign antigen. Everyone keep it in your mind. The MHC molecules are having two goals, ultimately having two goals. The first goal is antigen processing. What we can call the first goal? First goal we can call it as the processing, the most important process. Then the second one is the presentation. So first it has to process the antigen. When I use a word, listen my friends, when I use a word processing, you will understand. When I use a word processing, what you have to understand, we are breaking down the large antigenic peptide into smaller antigenic peptide. Why we have to convert large antigenic peptide into smaller antigenic peptide, friends? To present on the MHC molecules of the antigen presenting cells. What are antigen presenting cells? Like macrophages and dendritic cell. You're gonna see it very elaboratively. Listen to me very carefully. And very important and significant point. Without the help of MHC molecule, T cell never ever be active. It's highly impossible. Without association with the MHC, T cell never be active. Without association with the T cell, B cell never be active. If the B cell is not getting activated, friends, what's the point? You have to remember, there will be no production of the antibody only. So keep it in your mind. Just keep it in your mind. If the B cell wants to be activated, compulsory there will be a help of T cell as well as the MHC molecule. So class 1 and class 2 are related in both the structure and function. But if you look into the class 3 friends, every one of you keep it in your mind. We will be calling them as the group of unrelated proteins. Kindly do not get confused friends. Group of unrelated proteins which do not share any kind of a structural similarity also friends, as well as functional similarity also. First let me explain the structure briefly. Just pay attention, look into the diagrams very carefully and understand what exactly I am speaking. Listen to me carefully. This is, from here only we will expect the questions. This is structural differences only we got in the 4 marker question also, as well as in the 2 marker question also. Listen to me very carefully. There are, see, look into this. This is the structure of a class 1 MHC which consists of alpha 1, alpha 2, alpha 3 and beta 2 microglobulin. I want you people to understand this alpha 1 is a very large unit friends. Everyone pay attention, look into the diagram very carefully. This particular alpha friends, it is having a transmembrane domain also. At the same time, the most importantly friends, even it is having the cytoplasmic domain also. But friends, if you look into the structure of the beta 2 microglobulin, you can see the picture very clearly friends. It do not have transmembrane domain. At the same time friends, it do not have the cytosolic domain also. Kindly do not get confused with this particular point at all. Now all the students just listen to me very carefully. I have used one word. What is that word I have used? Listen to me friends, the MHC molecule has to present the antigen, right? So friends, everyone keep it in your mind. Whatever the area where the antigen is getting presented, everyone listen, that area we will be calling it as peptide binding cleft. Everyone, my friends, listen to me carefully. The area where the antigen is getting expressed, that is called peptide binding cleft. Friends, in case of a class 1 MHC molecule, the peptide binding cleft can be formed by the alpha 1 and alpha 2 subunits. I want you to remember this without fail. Okay, now look into the picture very carefully. Class 1 MHC molecule contain, everyone listen, 45 kilodalton alpha subunit and 12 kilodalton beta 2 microglobulin subunit. And what you have seen here, the alpha can be divided into alpha 1, alpha 2, alpha 3 subunits. Correct now friends? I want every one of you to understand a point that, listen to me, alpha 1, alpha 2, alpha 3 consist of 90 amino acid. Which domain? Extracellular domain. And 25 amino acids will make the transmembrane domain. Nearly friends, 30 amino acids will make the cytosolic domain. This is what I am talking about. Alpha 1, alpha 2, alpha 3. The extracellular domains consist of 90 amino acids. Transmembrane domain consists of nearly 25 amino acid. And 30 amino acids, what friends? They will form the cytosolic domain. And the most significant point, alpha 1 and alpha 2 only make the peptide binding cleft. Here one more very important and very significant point. When I use a word, listen my statements very carefully without confusion. When I use a word, we are expressing the peptide we are expressing the antigenic peptide. It doesn't mean that we can't express the entire antigenic peptide. Friends, peptide is a very large molecule. We can't express the entire antigenic peptide. So that's why we have to break it. After breaking, we can keep the antigenic peptide which is just in the range of 8 to 10 amino acids only. Friends, don't get confused. Suppose example, if the antigenic peptide is more than 8 to 10 amino acids, friends, we can't express them on the class 1 MHC molecule at all. Don't get confused with that point. Now look into the diagram very carefully, friends. This is the DNA, which consists of what? Alpha 1 chain, alpha 2, alpha 3 chain, transmembrane domain and cytosolic domain. Very simple. I am talking about how does the arrangement is occurring. These are all exams. Now it has given rise to the mRNA, same. Now this particular alpha 1, alpha 2, alpha 3 transmembrane domain, cytosolic domain, they finally translated into protein and they will be expressed. And to the alpha 1, alpha 2, alpha 3, who will come and associated, friends? Beta 2 microglobulin will associate. Beta 2 microglobulin majorly associated with what, friends? It's majorly associated with the alpha 3 domain. Kindly do not get confused with this particular point. And what I want you people to remember here, friends, don't confuse with my words. Beta 2 and alpha 3 domain, they resemble like immunoglobulin fold. I am not saying that they are acting like an antibody. No. I am just saying that their structure, when alpha 3 and beta 2 got associated with each other, they will look like immunoglobulin fold. Just they are looking, that's all. Now look into the class 2 MHC molecule. What does the class 1 consist of? Alpha 1, alpha 2, alpha 3 and beta 2. There is a little difference. What is that little difference? Look into the picture. If you look into the class 2 MHC, you can see, right, there is a presence of alpha 1 and alpha 2 chains and there is a presence of beta 1 and beta 2 chains. And please look into the diagram very carefully, friends. The alpha chains having transmembrane domain and cytosolic domain. Similarly, friends, the beta chains also having the transmembrane domain. At the same time, friends, cytosolic domain. Ankit, you will understand that question in the upcoming slides, but the question is very clear. That question is a valid question. You know, Ankit, you have to keep it in your mind. If the molecular weight is very low, undoubtedly antigenicity will be more effective. You know, right? If the antigen having the higher molecular weight, it cannot penetrate into the deeper tissues also. But when the molecular weight is low, the antigenicity will be more effective. That's a good point. You will understand in the upcoming slide. You have to understand that we are breaking the antigen into specific fragments which consist of 8 to 10 amino acids. That is more than sufficient to show the antigenicity property. Okay. Now listen, in the class 2 MHC molecule, friends, how does the peptide binding cleft can be formed? Everyone listen. In case of a class 1 MHC, what we understand, friends, the peptide binding cleft formed by the alpha 2 and alpha 1. Now every one of you listen to me very carefully, friends. In case of a class 2 MHC molecule, the peptide binding cleft is formed by the alpha 1 and beta 1. Kindly do not get confused with this particular point at all. And friends, keep it in your mind. In case of a class 2 MHC, in the peptide binding cleft, we can express the antigenic peptide which is in the range of 13 to 18 amino acids. This is one of the major significant difference between the class 1 and class 2 MHC molecule. There we can express only the antigenic peptide of 8 to 10 amino acid. Here we can express 13 to 18 amino acid. There the peptide binding cleft is formed by alpha 1 and alpha 2. Here the peptide binding cleft is formed by the alpha 1 and beta 1. And moreover, alpha also having transmembrane and cytosolic domain, and beta also having the transmembrane and cytosolic domain. Kindly do not get confused with this particular point. And look into the arrangement of exons, the same concept. This is the DNA, beta 1, beta 2, transmembrane domain, cytosolic domain. It has given rise to RNA. Now it is translated into the protein. To that, who comes and attaches? Alpha 1 and alpha 2 come and attaches. Point is very clear. Alpha 1, alpha 2 all are associating with each other by the disulfide linkages only. So listen to me carefully. So far whatever we have studied till this particular slide, if you did not remember, if you did not understand, just remember this particular slide and this particular table, more than sufficient to answer the question if they ask from the differences between the class 1 and class 2 MHC molecule. So pay utmost attention and try to understand what exactly I am speaking here. Same thing whatever we studied only. In case of a class 1 MHC molecule, the peptide binding domain made up of alpha 1 and alpha 2. Class 2 MHC molecule, it's made up of alpha 1 and beta 1. Friends, every one of you look into the second point very clearly. The second point saying that, listen everyone, nature of the peptide binding cleft. Everyone listen my statements very carefully, friends. Nature of the peptide binding cleft, listen to me, closed at both ends. But in the class 2 MHC molecule, it was written that open at both ends. Friends, every one of you look into the diagram very carefully, friends. This is the area whichever I am marking with red color. This is called as peptide binding cleft. Friends, if you look into the class 1, you can see the picture very clearly, right? On both ends, it got completely closed. But friends, if you observe the class 2 MHC molecule very correctly, look into the picture, there will be opening at both ends. Such a simple step it is. So the nature of the peptide binding cleft in case of a class 1 MHC molecule is it will be opened at both, it will be closed at both end, and in case of a class 2 MHC molecule, friends, it will be opened at the both end. Kindly do not get confused with that particular point. And I have clearly mentioned what is the general size of the bound peptides. I have very clearly told you that it consists of 8 to 10 amino acids. There is no doubt about it. And friends, when you look into the class 2 MHC molecule, we have discussed, right friends, understand we can express 13 to 18 amino acids. Now friends, just listen my statements. These two statements are very important, friends. These two differences are: focus, peptide motif involved in binding to the MHC molecule. Everyone listen, friends. Anchor residues at both ends of the peptide, generally hydrophobic carboxy terminal anchor. Anchor residues distributed along the length of polypeptide. Let me explain you elaboratively. Look into the diagrams very carefully, everyone. You only look into this, friends. This is the polypeptide, right? The blue color one is the polypeptide, and these are all amino acids. All of you look into the diagram very carefully. In case of a class 1 MHC, what is the statement, what is the diagram you can observe? You can observe only at the terminal position. Only at the terminal position, amino acids are associated with the peptide binding cleft. Yes or no? Yes. Then what about the other? Friends, every one of you listen to me very carefully. The other, the remaining entire polypeptide can be arranged as an arch. Can be arranged as an arch-like structure like this. You can observe it very clearly. Only friends, the terminal amino acids are only associated with the peptide binding cleft. But if you look into the class 2 MHC, what is the statement I can give you? The entire polypeptide, friends, listen my statements very carefully. The entire polypeptide associated with the peptide binding cleft, that is what they have given here. Anchor residues distributed along the length of the polypeptide. entire polypeptide associated with the peptide binding cleft. Here only the terminal positions only. Kindly don't get confused. So in the middle it form an arch-like structure, but here throughout the peptide binding cleft the arrangement can occur. Simple. These are all the major structural differences between the class 1 MHC molecule as well as the class 2 MHC molecule. Friends, right now what we have studied is just the structure. Now we have to study the regulation of the MHC molecule, and the most important one, that is the antigen processing and presentation. By chance, if you miss the questions from the structure, there is only one way that we can expect the questions from the what we can say that antigen processing and presentation. Sometimes in a given 4-marker question, they will raise the statements about the structure as well as about the processing also. So that's why you have to understand the concept very elaboratively, starting from the structure and presentation as well as the processing. Okay, first look at the regulation part. Just listen whatever the points I'm highlighting here. How this particular MHC molecule will be regulated. We all people know that inside the cell everything has to be regulated, because the regulation is the fundamental process to control the gene expression. So in every stage, in every cell, every process has to be regulated. Similarly friends, this particular MHC molecule also has to be regulated compulsory. Keep it in your mind friends. This particular class 2 MHC molecule will be regulated. How the class 2 MHC molecule will be regulated? It is regulated by two important molecules. One we can call the C2TA and RFX. C2TA and RFX. Just remember as it is whatever I said. These two are basically regulating the MHC, whereas MHC class 2. But friends, if you look into the MHC class 1, MHC class 1 can be regulated by INF, interferon alpha, interferon beta, as well as interferon gamma. Interferon alpha, beta, gamma are going to control the class 1 MHC. Along with this, even TNF also can regulate this particular class 1 MHC molecule. And friends, recently it was understood that, listen my words very carefully. I said that MHC class 1 is exclusively regulated by TNF alpha, beta, gamma, no doubt about it, and tumor necrosis factor. Recently it was identified that INF gamma, it is also regulating the class 2 MHC molecule. So in a simple way, what I can write? I can write C2TA, RFX and INF gamma. These three are going to regulate class 2 MHC molecule. Simple. Whereas INF alpha, beta, gamma and TNF is going to regulate the class 1 MHC molecule. Remember as it is, whatever I said. Okay, now the major part, which is the part we will get confusion. Antigen processing and presentation. See, I will tell you in a different way and in an effective way. I want you to understand me with a patience. You know, if I explain you 1 or 2 slides, you can't understand. If you understand the entire process, then only you will get a clear-cut idea about the processing and presentation. So what I am requesting you is understand all the slides. If with 1 or 2 slides you can't get the concept very correctly. And let me tell you in 2 lines, what is processing? Remember friends, processing means breaking down the large peptide into small amino acids. Simple point. Then presentation means we are presenting on the peptide binding cleft to help the T cell. Simple point it is. Now how we will do the antigen processing? Everyone listen to me, friends. Look into this. T cell require antigenic peptides to be displayed by the MHC on the antigen presenting cell. We shortly call it as APC. What are the examples of antigen presenting cells? We all people know very well. Macrophages. Even you can take the example of dendritic cells. Correct? Na? Now you listen, because T cells will take the antigen present on the MHC, then only T cell will release the cytokines, activate the B cell, then only B cell is going to give rise to the plasma and memory cell. Finally, there is a production of antibody. So what's the point here? It is compulsory and it is necessary, my friends. T cell require the antigenic peptide which is expressed on the MHC of the antigen presenting cell. Now you listen to me patiently. Antigenic processing of two types. One we can call, friends, endogenous processing, and another one we can call exogenous processing. Friends, kindly don't get confused, friends. I will explain elaboratively, just pay attention. Here you have to understand a concept very nicely. Exogenous pathway, keep it in your mind, mediated by the class 2 MHC. Endogenous pathway can be mediated by the class 1 MHC class 1 MHC molecule. First, in this particular slide, what I will do is just listen. In this particular slide, I will explain the overview how endogenous pathway will occur. Then the same process I will explain you step by step without having any kind of a confusion. Just I want you to listen to me very carefully. Then trust me, you will get the concept very easily. This is the endogenous antigen. Who will mediate this pathway? Class 1 MHC molecule. As I said to you very clearly, endogenous antigen, it's a very large antigen. I can't express this much large antigen. This is an overview. I can't express this much large antigen on the peptide binding cleft. So what will be the first step I should do? I have to break it into a smaller parts. Okay, now how this breakdown can occur? This breakdown can be mediated by a protein complex called as, friends, proteasome complex. Don't get confused. Now the large endogenous antigen I have break down into smaller peptides. Now the point is here, my friends, where the MHC molecule is present. Everyone listen to me carefully. The MHC molecule present in the endoplasmic reticulum. Now every student listen my statements. Now this antigenic peptides has to enter where? Has to enter into the endoplasmic reticulum, because they have to be expressed on the MHC, right? Where the MHC is present in the endoplasmic reticulum. So these all has to be entered into the endoplasmic reticulum. And the question asked is, how they will enter? Who is going to mediate their entry? Their entry can be mediated by an important protein, friends, which we can call the TAP protein, transporter associated with antigen processing. You will understand once again. What, friends? TAP. You always remember as TAP. Transporter associated with antigen processing. Now all the students can see the picture very clearly. The green color ones, which are the antigenic peptides, they are finally entered into the endoplasmic reticulum, and where they are expressing now, they are expressing in the MHC molecule. Such a simple step it is. Now once they express it on the MHC, you know what happened, friends? MHC, listen to me, friends, it will be packed in the form of a vesicle. What, friends? It is packed in the form of a vesicle, and it can bud off. Bud off means it can come out from the endoplasmic reticulum, and it will fuse with the Golgi complex. It will undergo the further processing. Finally, the vesicle will go and fuse the membrane. Finally, the antigenic peptide will be expressed on the MHC molecule. Finally, who comes into the picture, friends? T cell will come and take this particular antigen. This is just an overview. The same process we are gonna read elaboratively. Now you pay attention. All the students just look into the diagrams very carefully. So first, peptide generation is the first step. Now you only listen. What did I tell to all of you? This is the antigenic peptide. Friends, listen to me carefully. Focus. Now this antigenic peptide is a very large molecule, is it? So this much large molecule, we can't keep it on our MHC molecule. That's why this antigenic peptide has to be break down. Now the question comes here. How can we break the antigenic peptide? We can break the antigenic peptide by the proteasomal complex, which is a degradative complex. Now you all has to get a valid doubt. Sir, why the proteasome can come and degrade the protein just like that? No, that's not a simple process. You know friends, everyone listen to me very carefully. This is an antigenic peptide, right? This is epsilon amino group. This is an epsilon amino group. Friends, everyone listen. To this epsilon amino group of the antigenic peptide, ubiquitin molecule will come and attaches. What will come and attaches? Ubiquitin molecule will come and attaches in the presence of ATP. I told you many times, even in the plant hormones also we read, it's a general process. Whichever is added by the ubiquitin, friends listen to me, whichever is added by the ubiquitin molecule, that can be very easily degraded by the proteasomal complex. Now the concept is very simple, is it? Now the antigenic peptide associated with the ubiquitin molecule, simple. Now the proteasome is going to degrade this entire antigenic protein, antigenic peptide. Why? What is our ultimate goal? Our ultimate goal is breaking down into smaller peptides. Okay, finally we made it into smaller peptides. What is our ultimate goal now? I have told you very clearly. Now this antigenic peptide we have to take into the rough endoplasmic reticulum lumen. What will be the reason? Why I have to take into the lumen? What will be the reason? Because the MHC molecules are present in the RER lumen only. That is the reason I have to take it over there. So how is that possible? With the help of a protein called as a TAP1 protein. Now friends, I was repeatedly said that friends, in the class 1 MHC we can express only 8 to 10 amino acid. Is it or not? Why? Now this is the reason. That TAP protein, every student has to look into the statement. The TAP protein has the highest affinity only for the 8 to 10 amino acids only. That's why TAP protein can bring 8 to 10 amino acids only into the RER lumen. Finally, what should we do? We have to assemble this peptide on the class 1 MHC. Now this is the more important step, most important step. Pay attention, don't get confused. Listen my statements very carefully. We already read, right? The class 1 MHC made up of what? Alpha and beta. Alpha as well as the beta 2 microglobulin. Correct na? We already read it. Now every one of you pay attention, look into the diagram very carefully. Protein, by the proteasomal complex we break down into peptides. Everyone look into the picture. With the help of a TAP protein, it entered inside. There is no doubt about it. Now antigenic peptide entered inside. Now where it has to be expressed? It has to be expressed on the alpha and beta 2 of the MHC. Now please listen the statement whatever I am giving to all of you. Friends, in the absence of the antigenic peptide, which means antigenic peptide was not there. In the absence of antigenic peptide, the class 1 MHC is blocked. Class 1 MHC, alpha and beta 2 microglobulins are blocked by 3 proteins. What are the name of the 3 proteins that block the alpha and beta 2? Thapsin, calreticulin and calnexin. I want you to remember that. Thepacin, calreticulin and calnexin. These three are the proteins which are blocking the alpha and beta 2 microglobulin of the class 1 MHC in the absence of the antigenic peptide. Now you can see in this picture very clearly. Look into this picture, friends. This is the class 1 MHC molecule. It is associated with what? Look into this. Calnexin, thepacin and calreticulin. These three are the proteins blocking the MHC molecule. Now you only look into the picture very clearly. Now the antigenic peptide entered inside, which is brought by the TAP protein. Once the antigenic peptide comes into the picture, now you look into the diagram. Calnexin will be dissociated, thepacin will be dissociated, calreticulin will be dissociated. Now you can look into the picture. Class 1 MHC with the alpha and beta microglobulin, they will be expressing the antigenic peptide and they will exit the RER, processed in the Golgi complex, finally expressed where? On the membrane. This is what, friends? Endogenous pathway. I'll summarize in just 1 minute. Look into this. Endogenous pathway can be majorly shown by the class 1 MHC molecule. First, endogenous antigen at a particular amino acid added by the ubiquitin molecule with the help of a proteasomal complex. It can be broken down into small antigenic peptide which is in the range of 8 to 10 amino acids, because the TAP protein which is associated with the antigen processing, it is having the higher affinity with only 8 to 10 amino acid. Now the TAP protein bring the antigenic peptide into the endoplasmic reticulum lumen. Meanwhile, in the absence of the antigenic peptide, the MHC molecule is regulated by thepacin, calreticulin, calnexin. When the antigen peptide enter inside, thepacin, calnexin, calreticulin will dissociate from the alpha and beta 2 microglobulin. Now alpha beta 2 will join and antigenic peptide expressed over there and it will exit the RER, fuse to the Golgi complex, undergo the processing in the Golgi complex. Finally, they will come and fuse. They will come and express on the MHC. Immediately, who will comes into the picture? T helper cell will come into the picture with the help of TCR receptor. We can call it T cell receptor. They will take that particular antigen and they will release several cytokine molecules. Finally leads to the activation of B cells. That's a simple step. What pathway we are calling it as? Endogenous pathway. There is another pathway is also there, exogenous pathway. Every one of you please keep this point in your mind, exogenous pathway. Friends, don't get confused here. Exogenous pathway can be mediated by class 2. First let me explain you the overview. Once again I'll explain the step by step elaboratively. Friends, don't get confused. Listen my statements as it is. This is the class 2 MHC molecule. Listen my words. The peptide binding cleft of the class 2 MHC molecule blocked by a chain, inhibitory chain called as invariant chain. Once again listen. This is the class 2 MHC molecule. The peptide binding cleft of the class 2 MHC molecule blocked by the invariant chain. Okay, now this will be packed in the form of a vesicle and it will fuse the Golgi complex. Every student has to listen my words very carefully. In the Golgi complex, whatever the invariant chain that is blocking the peptide binding cleft, it will get digested. But friends, the point is here, out of 100% of the invariant chain, 90% of the invariant chain get digested and it will release it out. 10% of the invariant chain is left as it is on the peptide binding cleft. That is only we can call it clip, CLIP, clip. Now my friends, now you look into the picture very carefully. Exogenous antigen entered now. Now this exogenous antigen replaced this particular clip and expressed on the MHC molecule. That is what friends, exogenous pathway. Okay, so in the tomorrow class I'll explain this exogenous pathway step by step. Just it will finish it in 10 minutes. Okay, is it clear friends, everyone till here? Endogenous pathway, let me know everyone. I hope it's clear for everyone without confusion. And these topics are always confusing if you don't understand in a clear way. So I'm requesting all of you revise it once. Okay. And friends, we are having the class immediately 8:30 a.m. I'm sending you the link of developmental biology in the group. So please kindly attend without fail. Okay. So if anyone is unable to attend the 8:30 a.m. class, I am requesting all of you friends watch the recorded without fail. Okay, so please attend 8:30 a.m. class. I'll see you in the developmental biology class. Today is very important class because we are gonna start the cleavage. Okay friends, I'll see you at 8:30 a.m. Bye.